Okay, today we're going to change gears a little bit. I have voiced over, I like that word, chapter 22 slides. Chapter 22 in your textbook slides that you don't have the book for. But I think you can succeed with that. It's talking about pathogens in drinking water, water and foods. And so, and with that voiceover, we're going to have a few test questions that we'll formulate and put in. We have not put that up yet. So today is going to be the experiment on, we're going to try to isolate some of those pathogens that we're going to talk about in chapter 22. And one thing that I want you to take out of this thing, it could be a frightening subject to you if you are germophobic, but let's hope not. We're going to try to isolate some E. coli pathogens, and we're going to try to isolate Salmonella and Staph aureus, and those four organisms are in the top. They're the top four. There's 6 organisms at the top 4 of infectious diseases in foods. So we're going to go after 4 of them. The one we're not going to go after is called Campylobacter, and the only reason we're not, it is probably the most common foodborne pathogen, and it especially likes chickens and turkeys. But it's not limited to that. It also likes milk, and so the reason we're not doing it, it's pretty gnarly procedure, and I am missing one antibiotic, and I have the media to do it. I may play around with it later, but Campylobacter is the #1 food pathogen out there. It is a psychrophile. It grows in your refrigerator, and that's how it can infect your milk and so forth and so on. It does a pretty good job of it. And it colonizes humans' intestinal tract and chickens' intestinal tract. And when in the cecum, you have about 1 billion cells per gram. So that's a huge number. Think about it. So in the chickens, when you bring them to get plucked and skinned and put in a bag, you're taking the feathers off, you're gutting the animal, you're throwing them in a common bath, and these Campylobacters and other pathogens cover the outside of the chicken. So we're going to kind of use that as a enrichment for our little project today. So what we're going to do, we're not going to blend this chicken. We purchased one at a local grocery store. It don't matter where you purchase it, it's going to have the same percentage of these all the time. You're going to expect Staph aureus, you're going to expect Salmonella, you're going to expect Campylobacter. And what they tell you now is when you buy your chicken, the only thing that you need to know is that rascal's infected. So we're going back to this, how do you handle this contaminated bird? And first of all, you don't wash it in your sink. If you've got a smaller stainless steel something, you put it in. When you take it off, put the bag in the garbage, and then you can get your pan out. You can transfer the chicken to the pan, and you can salt and pepper it and all that stuff and zip into the oven. So the way we're going to do it, since we know that this organism has come from its intestines and it kind of goes over the whole thing, we're going to use a procedure of we're taking sterile phosphate buffer, and we're going to put it in this Ziploc bag. We're going to drop this chicken in there. We're going to shake our chicken till we think we got all the stuff in it, And then we're gonna either put it back into here, I'll figure it out as we do it, and we're gonna go over some of the selective enrichment media that we're gonna be setting up to isolate. So with that said, this is our phosphate buffer sterile that we're gonna pour in here with the juice that came from the chicken itself. And I don't want to get too much. I'm just gonna go maybe, let me see what we're doing. Need a little more. I want to completely cover it. Now let's hope we don't have a plastic breakdown.
Blowout.
Blowout. This should contain it. And so at this point, I'm still going to hold it because I don't trust it. I'm gonna wash all around, make sure that I get inside the bird. And as you notice, we're getting darker and darker as we're doing this, which means we're washing off stuff that's getting in our water. Now, as you can tell, we've got chicken stuff everywhere, but that's okay. I want to get all the juice. And my notes have moved over. Now be filtered with how we doing? All right. I want to save this somehow or another. So I'm going to put this, we're going to bake this chicken, not right now. We had big plans, but that kind of got altered. So it's going to go in the oven tonight, and I'll let you know how that works. Okay, here's our wash of freshly plucked chicken. And what we're going to do is we got several selective differential media that we could use. This doesn't cover all of the types of media that's out there. It covers what I have been successful with. We're going to start off, first of all, we're looking for coliforms. The wild strain is not pathogenic, but we are looking for the strains that have picked up toxic genes. So this is EMB, eosinmethylene blue. If you had 224 or 205, you have seen EMB before. It is selective for gram-negative. It has bile salts in it, and the eosin and the methylene blue are inhibitory to gram-positive organisms. So that makes it selective for, and the main thing it has in it is its differential. It has lactose. Glucose is a single sugar. Lactose is a disaccharide. Anybody remember the two monomers in lactose? Glucose and galactose. Okay, you're off the hook there. Now, so we're going to when we go to test this juice, we're going to do it two different ways. We're going to basically take a loop, fire a loop, and we're going to streak a couple of these. And a couple of these, we're going to take 1/10 of a ml, we're going to put on here, and we're going to do a spray plate. So let's do one of those, and I don't think you need to watch the whole thing, but let me go through and cover the different selective and differential media for you to start with. We will put all of this information in somewhere, a protocol, or we talked about doing another, when we see the results, we'll actually film it, and at the time we film the results to talk about what we got, then we're also going to give you that information about this different media that you don't have to, we're not going to burn you out on the selective agents, but I'm going to go over a couple of them. So the EMB is for the E. coli pathogens. The Staph aureus is one of the top 4 organisms that's leading in infectious diseases in foods and water. We have 2 different media that we can use here. Staph aureus, if it's coagulase positive and hemolytic, one of the ways that we can differentiate staph is there's another organism that is not pathogenic, Staph epidermidis, and one of them ferments mannitol, one of them don't. So what this has in it now is mannitol,
phenol red,
and phenol red's pH indicator. So if it ferments it, it's going to turn yellow. So we know that so Staph
aureus.
No, 110 I think is the name of this one, this media.
What?
Staph 110.
No, it's called MSA.
Okay, MSA. So we're going to streak and do it, and we can differentiate right off the bat with this, whether it's Staph aureus or Staph epidermis. But that doesn't tell you, it doesn't confirm that it's Staph aureus, pathogenic Staph aureus. So, but this will strictly tell with fast whether we got a Staph epidermis or Staph aureus. And chickens are no different than us. It gets on the Staph epidermis, can get on the skin also. It's just not a pathogen. So the other one we have is my favorite one because it's got a bunch of gooky stuff in it. This one's called Beard-Parker agar. And it's a selective media, and it's selective for coagulase active staphylococci. That is a prelude to saying it's pathogenic. It has 2 things in it: lithium chloride. Anybody familiar with lithium? Lithium, it's a heavy metal, lithium chloride. Potassium tellurite are selective agents for Staph aureus. Tellurite is going to make the colonies, if they grow in presence of tellurite, they're going to turn black. And if they are going to look like something called egg yolk, that's the other thing, lecithin. And it's going to tell you about lipase activity, it's going to tell you about lecithinase activity. Both of those correlate with virulent Staph aureus. So we're going to streak this plate, and we're going to get some black cells with a halo, and we're going to do a coagulase test. Not with this, we're going to take a colony and do a coagulase test, and then we're pretty much sure that it's Staph aureus. That's a lot of babbling for one little thing. And it makes a neat colony that you can look at. So this Beard-Parker is going to be confirmation of Staph aureus. This one just tells you it's either Staph epidermis or Staph aureus. The last one we're going to talk about, and I'm going to do this one a little quicker, this one is called bismuth sulfite agar. Bismuth sulfite agar. This is for salmonella. It was developed primarily for Salmonella typhi, but we don't have that much no more, but it's also good for all the 1,500 serotypes that cause gastrointestinal diseases. And the beauty of it is, it's got the bismuth sulfate. Salmonella, one of its giveaways is that it can produce H2S. It does it in your system as a way of invasion, but on this agar, it's going to turn, the colonies are going to turn black. So we got a black colony over here because that, when happens when you produce H2S and you got iron in it, it turns black. And they call this a rabbit eye colony. And if you, when you look at it on the plate, those colonies, see the dark center and the lighter, that's what a rabbit eye looks like. That's country people for dictating bismuth sulfite. So we are going to tell you more about this when we see the positive results. So I'm going to do, one of the things we're going to do, and this is, we don't need to watch all of it. We're going to start off and do this is quicker. And so all we're doing is we're letting that cool a minute, and we're going to see if there's any E. coli in here. We just streak that across that way. I can turn this over and streak another plate. And I can use the same loop for all the streak tests. So it doesn't make any difference. I'm going to streak all of these, and you don't have to watch me do the spread plate. Okay, we just finished checking a chicken, and we did a number of plate counts and buffer water where we shook it up on the slide, and we tested for E. coli, and we tested for salmonella and staph. So remember, on the staph we did 2 different types, and I think we can look at this, and if you turn this over, I think you could see the yellow halo on the positive Staph aureus. This media is distinguishing between Staph epidermis and Staph aureus. So Staph aureus is positive. So if you were to count the positive colonies, it's around 10, and we did duplicate. Remember, we put 1/10 ml of that rinse onto this, and this is the second plate. This one actually has got better halos than the other one. I think you can see those. They're pretty wide. And there's about 10 colonies on this one. So if you average the count, you had 10 colonies per 1/10. So that means 100 mLs, 100 staph colonies per ml of that rinse water we had, if you want to look at it like that. If we want to now, this just tells us the difference between Staph epidermis and Staph aureus. Now the next thing we're going to have to do, we set up another media called Beard Parker with lethosinase in it, and on that one the staph will come out black, and you can see some halos. This is kind of a young, early, this probably needs to incubate a little more to show the coagulation, but you can see the black color would be staph. And the only thing left for us to do now to confirm this is to take a gram stain, of course. We'll take of this, and then we will take some of this and do a coagulase test. So if it's positive for coagulase test, then we have the pathogenic Staph aureus on that chicken. So that is two examples of what we could do there. The bismuth sulfite is for salmonella. There's a number of serotypes of salmonella. We have the composite polyvalent Anacera that we can test, and these are still a little young. So if they incubate a little more, they would be black. We have restreaked these, and so we're going to do maybe a polyvalent test on it. If I can find my stuff in the refrigerator, that would confirm what group of salmonella was on there. So we still have to confirm all of this. And the E. coli, just to give you an idea of what's in a 10th ml, that glob right there and we are going to restreak some of these colonies. You can maybe pick them out that it looks like E. coli, and we're going to restreak those and confirm that E. coli. So of all the potential pathogens to review, we do have some Salmonella, we do have some Staph aureus, and we don't know for sure if we any of the E. coli. So that would be where we stand on food pathogens and the chicken